Hello, dear students. I hope you all are fine and doing well. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear students, we have already started with a lecture series of sexual reproduction in flowering plant. In that, in previous lecture, we have studied about the endosperm and embryo. Dear student, you know that these both structure are formed within the seed. So we have to deal with the seed part as well. So if we talk about the seed, you know that seed is actually derived from the ovule. So we will be talking about this seed in detail. And in that, if we talk about seed, you know that it is often described as a fertilized ovule. Dear student, it is described as a particularly fertilized ovule. And this fertilized ovule means the ovule is getting transformed into seed. And this ovule is covered by ovary wall, and ovule and ovary wall forms a ovary. And dear student, if we talk about the transformation of ovary, ovary convert and form fruit. But we are going to talk about mainly seed in detail. So this seed is nothing but converted or derived by the transformation of ovule. If we see a typical seed, in seed you will find a seed coat, cotyledon and an embryonal axis. Dear student, already we have studied this part in previous lecture as well. Here you can see the seed coat is there, cotyledons are there, and embryonal axis is there. See, this part is a cotyledon. This part is a seed coat. And this part is representing the embryo. Okay. Which can be shown here detail. Here you will see the particular, this is the embryonal axis. These are the cotyledons. Okay. These are the above some leafy structures which is showing a particularly developing embryo. Okay. Now we will talk about this structure in depth as well. And in this we will be dealing about the dicot seed and monocot seed separately. Now we are talking mainly about the structure of dicot seed. In dicotyledon seed, two cotyledons are present in the embryo. Because of this seed are known as dicotyledon seed. And this dicot seed also have an outer covering of the seed that is known as seed coat. But this seed coat is two-layered or bi-layered. Dear student, be remember, the seed coat is bi-layered. They have a two-layer, in which outer layer is known as testa and inner layer is known as tegmen. And actually this outer testa is a transformation of outer integument of ovule. Means outer integument which is found in the ovule get converted into the outer testa, and the inner integument of ovule is transformed into tegmen. So when we see the structure of ovule, we have already seen that in ovule a structure named hilum is found, which connect the funicle with the body of ovule. Same way, hilum is also found here in case of seed as well. And here, this hilum is represented by a scar. It is represented by one scar on the seed coat, and that seed coat should be the outer one, and that is the testa. And through this scar represent a structure through which the developing seed were attached to the fruit. Means If a seed is present within the fruit, then through this scar it is known that it can be determined that from which point the fruit is attached with this seed. So you can see here, these are the seeds, and it is beautifully shown in this diagram that this is a seed, and you can see in this particular seed a scar here. Okay, so this is a hilum. Okay, now this is also one of the important structure of bean is shown here. It's a seed detailed structure, and in that also you can see here particularly the outer. You can see here the seed coat is there, outer and inner. This is the two cotyledon part, this is the cotyledon plant, and this is the particularly attachment of the part where a funicle can be observed, there's hilum can be observed. Now in a seed above the hilum, small pore is observed which is known as a micropyle. If you see, if suppose this is a seed coat, and if it has a scar here, then pore is observed here. Here you will find a scar, and here you will find a pore. This pore represent a micropyle, and this pore is usually help seed for the exchange of gases and entry of water. Okay, so within the seed coat, the embryo consists of embryonal axis and two cotyledon. You know that the cotyledon are often fleshy and full of reserve food. Yes, they have ability to store the food material. So you can see this is the part which is showing a particular structure of a dicot seed, and in this seed you can see these are the particularly cotyledons, and these cotyledons are rich in food. They have a seed coat and they have an embryo which is also known as baby plant, because this is going to form a new plant. Okay, so dear students, this is a detailed structure of a dicot seed. Now at the two ends of embryonal axis are present a radicle and plumule. Yes, dear students, we have seen this diagram particularly in case of dicot embryo, and we have shown this is the embryonal axis. Here you will find a cotyledon, and here you will find a plumule. Here you find a radicle. Okay, so this statement is saying that two ends of embryonal axis are present. One is the radicle and one is the plumule. Now in seed coat you can see one detailed diagram here again, and in this we can see the seed coat is there. Here two seed coat is there, outer testa, inner integument. This is the plumule. This is the plumule. Okay, where below which cotyledon is attached. This is the hypocotyle, means it is this part. The region of embryonal axis between cotyledon and radicle is known as hypocotyle. So this is showing hypocotyle. Here you can see the hilum. Here you can see the micropyle pore, and these are the cotyledons. Okay, such a way they are arranged. Okay. Now, dear student, if we go on talking about this in further detail, we'll come to know that this is also one of the important diagram shown here. You can see that here this is the shoot and this is the root. See, this is the shoot, these are the cotyledons, and this is the root. Now here this root is arised from the radicle, this shoot is derived from the particularly plumule, and this seed coat is seed will be released from this particular plant, means from the seed this particular whole plant is seed germinated. Now this detailed structure we have studied. We have also studied about the hilum, which is a scar. Next to that, in some seed, such as castor, the endosperm formed as a result of double fertilization is a food storing tissue. Yes, students, we have already studied about this. Endosperm is a food storing structure, and this food storing structure is found in the seed. Okay, so we know this endosperm, and it is formed as a result of double fertilization. Yes, it is found in the double fertilization. It is formed specifically by the triple fusion, and so we know that this endosperm is formed by the triple fusion, and if it is formed by a triple fusion, it is triploid. Next to that, you can see this is a castor, and we have studied already about the oil of this particular castor, and they have an oily endosperm. Apart from that, in plants such as bean, gram, and pea, the endosperm is not present, and they are known as non-endospermic seeds or non-albuminous seeds. That also we have discussed in the previous class. Now, after this, we are going to talk about the structure of monocot seed. And in structure of monocot seeds are known to be endospermic, but some in orchids are non-endospermic because orchids do not have a particularly endosperm. They are non-endospermic seeds. When we see the structure of maize, the seeds of cereals such as maize, the seed coat is membranous and generally fused with the fruit wall. One of the important feature of the particularly in maize, whatever the seed we eat actually, that is not a seed only, they have a fused pericarp as well, or they have a fused fruit wall as well. Okay, so here this is one of the important point that you should remember. Seed coat is membranous, but seed coat is generally fused with the fruit wall, and whatever we eat, not only seed but we eat fruit wall as well. So you can see in this is not showing just a particularly, this is not showing only a seed, but in this the particularly pericarp is also fused, you can see here. And this is the particular diagram of internal structure, but internal structure of monocot embryo, monocot seed we are going to discuss. So these are the orchids. This is the maize. Yes, we have to study maize because mainly you get a question with respect to the maize. And when we talk about the maize, we come to know that, dear student, this is the actual structure of maize. Now in maize, you will find the endosperm. This region is representing the endosperm. Okay. And this endosperm is covered by a membranous layer that is known as aileron layer. We have studied. And this aileron layer, here endosperm is triploid. Same way aileron layer is also triploid. And this aileron layer is proteinaceous, or it is rich in protein. That also we have studied in previous class. Now here this is showing the remains of style. Okay. Here this is the showing important structure, that is what? Fused pericarp and the seed coat. Then here you have one important, this part, that is cotyledon. This cotyledon is known as scutellum. Then we have a coleoptile, which is having some small leaves which is covering the plumule. So coleoptile is covering the plumule. Then radicle is covered by the particularly root cap and coleorhiza. And the layer which differentiates this endosperm from the scutellum, that is a epithelial layer. You can see here this epithelial layer is given. So this is all about maize or monocot seed. Okay. So let's talk further. Now, if we talk about endosperm, endosperm is known to be bulky and it has a main function of storage of food. And the outermost covering of endosperm separate the embryo by a proteinaceous layer called aileron layer. Just we have seen in the diagram of maize that aileron layer is proteinaceous. Okay. So the proteinaceous layer, this question is most important. They usually ask question from this part. Okay. So that is important one. Next to that, you can see the natural diagram of this one, in which you will see the endosperm, pericarp, and germ part of this particular seeds. Then within the seed you will find an embryo and endosperm, where embryo is small and situated in a groove at end of the endosperm. Dear student, we have seen just a diagram of this particular maize seed. And in maize seed, when we seen a diagram like this, we seen the particular arrangement of embryo like this, and whole seed is present. So this is the embryo. Means this embryo is a small one and situated at a groove at one end of the endosperm. Okay, because here we have seen the presence of endosperm. Okay, here we seen the presence of endosperm. Next, it consists of one large shield-shaped cotyledon known as scutellum and a short axis with a plumule and radicle. Yes, we know that scutellum is a single cotyledon of this monocot. Plumule radicle are also enclosed in a sheath which are called coleoptile and coleorhiza respectively. Coleoptile cover the plumule and coleorhiza cover the radicle, that we have seen. Now, the significance of fruit and seed is that the integument of ovule harden as tough protective seed coat. You know that integuments are modified and form a what? Seed coat. So we have studied till now that integument modify and form seed coat. Outer one form a testa and inner one tegment. We also know that ovule completely modify and form a seed. The pericarp or particular edible part of this or fleshy or dry part of the fruit is formed by the conversion of ovary wall. So ovary wall convert and form a pericarp. Okay, so this is the conversion. Now when we talk about the particular outer seed coat testa that develop from the outer integument, just we have discussed. Okay, we have just discussed this. And here testa, you can see that testa develop from the outer integument and tegment from the inner integument. Next to that, we come to know that micropyle remains as a small pore in the seed coat which facilitate the entry of oxygen and water. You can see that this particular detailed diagram here. You can see these are the testa and tegment. Here you can see this is the endospermy part, and like this particularly seed is observed. But this diagram is not much important for us. We have to talk about this. Micropyle is a seed pore actually that facilitates the entry of oxygen and water into the seed during the germination. And when oxygen and water availability occurs, then only the embryo will germinate within the seed. Next to that, when a seed matures, the water content is reduced and the seed become relatively dry. So when a seed matures, the water content is reduced. So the water content become 10 to 15% moisture by mass. And you can see these are the particularly germinating seed where they are started germinating or coming out of the seed coat by breaking a seed coat. The embryo may enter a state of inactivity. Sometimes what happens, the embryo do not germinate and it remain inactive, and that inactivity state is known as dormancy. So this is particularly we can say a dormancy. Or if favorable conditions are available, adequate moisture, oxygen and suitable temperature, they germinate. Okay, so this is the what particularly dormancy. You know that dormancy is a sort of state of inactivity. So when unfavorable conditions are there, the dormancy can occur. Sometimes dormancy, there can be a different regions of dormancy. You know, dormancy can occur due to different regions. So first we talk about regions of dormancy. And when we talk about dormancy regions, the first region is hard seed coat or mechanical seed coat. Second, impermeable seed coat. If seed coat is impermeable due to impermeable to oxygen or water. Third, there can be a possibility of immature embryo, lack of growth promoters or nutrition. This all can be the possible reasons for the what? A dormancy. Okay. Now, when a seed is there, what seed is responsible for the generation of new life? So seed dispersal can also occur. And when we talk about a seed dispersal, a fruit is considered as an adaptation for a dispersing seed. Coconut floats, Martinia, tiger, clue plant is there, that can also float. So due to this mechanism, the seeds are travel with the fruit and they disperse and reach to the other part. Now, fruits also develop different mechanisms for the dispersal of seeds. You can see these are the different mechanisms through which the seeds can be traveled to another place with the help of fruit. You can see in this milkweed, you can see this is the hairy structure is present, and due to this hairy structure, this is the hairy calyx due to which this can move. Here also this is the hairy calyx which help them to move through the wind. Here you can see this particular things help them to move. So by wind also the seed can travel to another place. By animal, these baguette ticks are there, sandbur are there, blackberries are there. Okay, these all are particularly travel to the which one? By animals. By water, lotus seeds, then cattail seeds, coconut, they all can particularly travel or disperse by their seeds are dispersed by the water. Sometimes seeds are also released by bursting. You can see in violet the particular seeds are bursting. Then by humans also, like bean, wheat, cherry, these are particularly say travel by the humans. Okay, sometimes we eat a fruit and we throw a seed nearby. Then due to these regions, the seed can disperse to the different places. Wherever they find the suitable conditions, they will germinate and they will give rise to the new plant. Now, from that, we have to talk about the different sizes of the seed. And when we talk about different size of seed, we come to know that there is a wide variation in the size of mature seeds. There is a wide variation. In this wide variation, if we take example of orchid, the seed is microscopic. The seed are microscopic. And the seeds are produced in more numbers. Each seed matures 85 micrometers, measures 85 micrometers, and weight is 0.181 micrograms. This is the particular size of orchid seeds. But this size need not, there is no need to remember, just be remember orchid seeds are microscopic. There is just too small size. Some larger seeds are also observed in the Laudacia maldivica, the double coconut. Its size, if you see the size of seed, you can be surprised that this is 30 centimeter long seeds are found. And nearly 90 centimeter in circumference and 18 kg in weight, which is showing that there can be a large size seeds are also found. Then this is a particularly seeds of Laudacia maldivica. Next to that, there are some advantages of seeds to angiosperm, dear student. If we see, seeds have better adaptive strategy for dispersal to a new habitat and help the species to colonize in another area. Suppose if this area become unsuitable for the particular plant, then there will be a better area for the seeds in a different area. So it is particularly possible that they can have a different adaptive strategies for the new habitat, and there they can populate the area of that particular species. You can see here, this way different seeds can be released. They can be travel through air, they can be travel through water as well, and colonize a new habitat. Some birds also can help in this particular condition. Then, till they have a sufficient food reserve, the young seedlings are nourished until they are capable of photosynthesizing on their own. Young seedlings also get nourishment from that only, reserve food only. But when they gain a capacity of photosynthesis, they will be nourished by themselves. So this is the initial saplings which are not photosynthesizing, but they have a particular storage food and they are gaining the capacity actually. Hard seed coat are that we have seen that due to the hard seed coat also embryo cannot come out of the seed. Apart from this, hard coat also provides some beneficial mechanisms, like they provide protection to the young embryo. And seed, as being a product of sexual reproduction, they generate new genetic combination leading to variation. Means the plant which has produced seed and the plant which is generated newly from that seed both have a different genotype. And this is the one particularly structure of seed. We have seen so many diagrams of seed till now. So seed forms the basis of agriculture, because seed is going to germinate, seed is going to form a new plants. So dear students, we have just seen the term dormancy as well. Now seed dormancy, if we say, then it is known that dehydration, that is a loss of water, and dormancy of mature seed, that is a suspended growth, these two things are crucial for the storage of seeds. Because if we need to store a seed, then we have to stop them from germinating. and they can be stopped only when they particularly have a dehydration. And these two characteristics help them to store, and so they can be used as a food throughout the year. And if there is a need in the next season, we can grow them, raise them, and get a crop in the new season. So now this question sometimes you will get in a blank form of you. So you should remember these two terms. These are the crucial for storage, and in NCERT also this is given. So important one. You can see this is the storage part. And if we see about the particular these seeds, some seeds lost their particular viability within a few hours or few days, actually few days. But there are several records of very old yet viable seeds. Means they are so much years old, but still they have ability to germinate. And if we see this type of particular seeds, then the oldest one of this is the lupin. Oldest one is the lupin. And if we see lupin, that is a Lupinus arcticus that is excavated from the Arctic tundra. Okay. This seed germinated and flowered after an estimated record of 10,000 years. Dear student, this is the data which you have to remember. You get questions so many times from this part. Okay. So you have to remember this 10,000 years of dormancy of which one? Lupinus. Okay, that is the important one. And so many times you can get a question from this part. Okay. Apart from that, the recent record of 2,000 years old viable seeds have also obtained of a date palm. This is also one more data. 10,000 years we have seen. This is another data of 2,000 years old. And that is a date palm. And that is Phoenix dactylifera. And this Phoenix dactylifera date palm seeds discovered during the archaeological excavation of near King Herod's palace near a Dead Sea. So be remember the 2,000-year-old sea viable seeds obtained during the excavation from the King Herod palace. And this is a particular date palm which was observed there. So date palm have how much? 2,000 years of viability. And this viability is the ability of a seed to retain the power of germination. And it can be tested by using a 0.1% solution of triphenyl tetrazolium chloride. This triphenyl tetrazolium chloride particularly help us to study the germination power or the viability of different seeds. Some orchid fruits are that they contain thousands of sign seeds, similar in case of fruits of ficus and parasitic species such as Orobanco and Stiga. Means they have multiple seeds in a fruit. But there are so many fruits are there which have a single seed as well, like mango. But here they have a particular multiple seed. So you should remember this example. Sometimes they ask in which multiple seeds are particularly found. Then let's talk about some of the questions. That seed is described as a fertilized. You know that it is modified from the ovule. So seed is defined as a fertilized ovule. So the answer is ovule. Next question is outer layer of seed is called. Yes, you know it very well. And we have studied just before few slides. It is a testa. And with respect to that, the eggman is nothing but an inner seed coat. So answer is testa. But what are the other terms? You know, hilum is a scar, okay, which represents the attachment site between the seed and fruit. And a raphe is also found. We have studied this raphe during the what? A particular structure of ovule. Then the third question is, seeds of which plants were discovered during the archaeological excavation at King Herod's palace near the Dead Sea? Dear student, this also we have studied just now, and you know that it is the seeds of 2000-year-old Phoenix dactylifera, that is dead palm. The next question is, the viability of seed can be tested by using, just this also we have discussed, that is a particular 0.1% solution of TTC. The portion of the embryonal axis above the level of cotyledon, dear students, we have studied that a region of embryonal axis above the cotyledon is known as epicotyle and below the cotyledon is known as hypocotyle. So the answer should be epicotyle. Hypocotyle is below the cotyledon. What is coleorhiza? Coleorhiza is a protective covering of radicle. And radicle is the structure of embryo which has the ability to form a new roots. Okay, dear students, so till now seed part is over. Now we will be dealing with the one of the important topic, and which is the last topic of this chapter, that is apomixis. With that apomixis, we will also study the parthenocarpy. We will also discuss about parthenogenesis, and at last we will be talking about polyembryony. So dear students, after studying this many things, we are at the verge of this chapter, and we will be dealing now a new interesting topic, a very interesting topic. Dear students, why this is a very interesting topic? Listen. Till now, whatever we have studied about sexual reproduction, and in sexual reproduction, we have talked about the meiosis, which forms a male gamete and female gamete. Then we studied about the syngamy or fertilization. Then we studied about the formation of zygote, and zygote formed an embryo. Such a way sexual reproduction has occurred. An embryo formed from zygote, and before zygote, fertilization and meiosis happened. But dear students, apomixis is a important type of event where you can see there is a no meiosis, there is a no fertilization, but still the embryo is going to be produced. So it is something different. So let's talk about this. Apomixis. So apomixis is a formation of seed without meiosis and without fertilization. So if we define them, that is known that all those seeds in general are a product of fertilization. A few flowering plant, such as some species of Asteraceae and grasses, have evolved a special mechanism to produce seed without fertilization. So dear students, be remember apomixis is a production of particularly produce a seeds without fertilization. And in this, it is found in two species, that is Asteraceae and grasses. Okay. So now, this is the apomixis or agamospermy. So apomixis is a form of sexual reproduction. It is a form of sexual reproduction, but it mimics sexual reproduction. It is a form of asexual reproduction. Actually, it is not written here, but it is a form of it is a form of asexual reproduction which mimics sexual reproduction. Means it is actually a asexual reproduction. And there are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reductional division and develop into the embryo without fertilization. Yes, without meiosis, the diploid egg will be produced, and this diploid egg is going to form a embryo. Then that is without fertilization, that is a part of apomixis. Same way, and we will study that this thing of formation of diploid egg cell and which have developed into diploid embryo without fertilization, we will talk about this is as diploid parthenogenesis. Okay. It is an assured reproduction in the absence of pollinators, such as in extreme environments. And here we are going to talk about a different type of this apomixis. So let's talk about the different types of apomixis. Okay. Now, if we talk about this apomixis is also known as agemospermy. And here a formation of seed occur without meiosis. This agemospermy occurs within the seed. Occur within seed. And here embryo are also produced by this asexual reproductive method instead of normal sexual method. And this is of different type. And in this we are going to talk about that it is of three types. Number one is recurrent agemospermy. Which one? Recurrent agemospermy. Now, in recurrent agemospermy, the egg is diploid as the embryo sac is formed directly either from the nucellar cell or from the diploid megaspore mother cell. And the diploid egg developed parthenogenetically into diploid embryo. So suppose here I am showing you diagrammatically. This is a ovule. You know that this is the nucellus. And suppose one of the nucellus cell get modified and known as megaspore mother cell. You know that nucellus is also diploid and megaspore mother cell is also diploid. Now if this diploid nucellar cell or this diploid megaspore mother cell undergo division and form a diploid embryo sac. These are the synergid. This is egg cell. This is polar nuclei. And these are antipodals. And these antipodals are present within the these secondary nucleus are present within central cell. See, So if this is a diploid cell, it undergo mitosis and form a structure, then diploid synergy will be produced. Egg will be also diploid. Antipodals are also diploid. Central cell, which having a two polar nuclei of 2n each. If this diploid embryo sac is formed from the diploid nucellus, then it is known as somatic apospory. It is known as somatic apospory. And if this embryo sac developed from the diploid megaspore mother cell by mitosis, then this is known as diplospory. Or generative apospory. Now, so these two things can be defined as formation of diploid embryo sac from the diploid nucleus nucellus is known as somatic apospory, whereas generative apospory or diplospory is formation of diploid embryo sac from the diploid megaspore mother cell. Now, either this can happen or this can happen, but diploid egg will be produced. Now, if this diploid egg undergo mitosis and produce diploid embryo, then this is known as diploid parthenogenesis. Okay, so these both are the type of recurrent agamospermy, and this thing occur without fusion of gametes, without fertilization, and such a way diploid embryo can be obtained. Such type of thing, apomixis, is observed in case of apple. Okay, so this is recurrent agamospermy. After recurrent agamospermy, we are going to talk about another type, that is adventive embryony. And in adventive embryony, you come to know that here embryo developed directly from the diploid cell of the ovule lying outside the embryo sac, like nucellus or integument. So here we also have to draw a particularly ovule. So in this, you know that this is a part of funicle, this is hilum, this is outer integument, this is inner integument, and this is nucellus. Now this is also diploid, this is also diploid, this is also diploid. So out of this two, integument or nucellus, undergo division and without forming embryo sac, if they directly give rise to the embryo, then it is known as adventive embryony. So here what happens, here embryo develops directly from nucellus or integument. Whereas in previous recurrent angiospermy, what we have studied, first embryo sac will be formed, then embryo will be formed. But here what is happening? Direct embryo is getting formed from the nucellus or integument. Now this adventive embryony, if you talk about example, then is found to be seen in case of mango. It is found to be seen in case of onion, then in case of citrus, opuntia. Okay, and it is known that in citrus there is a presence of 2 to 40 embryo. Means multiple embryo are found. So it is an example of polyembryony as well. So we will study, and this adventive embryony, dear student, is also known as sporophytic budding. Okay, so this is the part of adventive embryony. Okay, so two type we have discussed. Now we are going to talk about another type, that is non-recurrent agemospermy. Now, when we talk about this non-recurrent agemospermy, in this type, what happens? The megaspore mother cell undergo normal meiotic division, and one of the 4 megaspore thus formed develop into haploid female gametophyte, that is embryo sac. Means in this, normally haploid embryo sac is produced. So this is a embryo sac or female gametophyte which is haploid. These are synergid. This is egg cell. These are antipodal. And it is produced normally by meiosis from the megaspore, which is produced by the meiosis of megaspore mother cell. Now in this, no fertilization takes place, but embryo still develops from the unfertilized egg cell. Suppose this is egg, so it is an unfertilized egg. Now this egg undergo mitosis and produce an embryo. Then this process is known as haploid parthenogenesis. So if we need to define, how can define this haploid parthenogenesis? It is a formation of haploid embryo by the mitosis of haploid egg. Okay. Apart from this, from the some other cells of the haploid embryo sac, if the embryo develops, then it is known as haploid apogamy. So next is haploid apogamy, and in this, what happens? Embryo is produced by division of cells of embryo sac other than egg cell, means from the cells of antipodal or synergid. If the embryo is produced, then this embryo is going to be haploid, then it is known as haploid apogamy. But in seed plant, such embryo are going to be haploid, and this haploid embryo does not survive. Okay, so which is the one of the important peculiar thing about this. And these are the three types of apomixis which we have studied here in detail. And now I hope you are clear with this apomixis. So the hybrid varieties of our food and vegetable crops are extensively being produced. Okay. Cultivation of hybrids has tremendously increased productivity. One of the problem of hybrid is that, what are the problems of hybrid? Then one of the problem with the hybrid is that hybrid seeds have to be produced every year. If the seeds collected from the hybrid are sown, the plant in the progeny will segregate and do not maintain hybrid character. Means hybrid character is going to be conserved for only one generation. It is not going to be conserved from generation to generation. Okay, so there is a need of production of hybrids every year. And the production of hybrid seeds is costly, hence the cost of hybrid seeds become too expensive for the farmer. So how the hybrid seeds will be beneficial or advantageous or disadvantageous, that can be seen from these particular points. If these hybrids are made into apomictic, there is a no segregation of character in the hybrid progeny. Now this is the important one, because in previous slide we have studied that what happens whenever there is a particular hybrid is produced, the hybrid will release the particular new variation in the next generation. It will not continue because the segregation will occur. But if we need to continue this hybrid character, then this apomixis will help, because in apomixis the meiosis is not going to happen. So if these hybrids are made into apomictic, there is a no segregation of character in the hybrid progeny and it will be continued. Then the farmers can also keep on using the hybrid seeds to raise a new crop year after year, and he does not have to buy hybrid seeds every year. They can produce the hybrid, and that hybrid can be used to produce a new hybrid plant by the apomixis, and that new hybrid plant will again give seeds. That seeds will be used for the production of again a new hybrid, and the particular thing will go on. So because of the importance of apomixis in hybrid seed industry, active research is going on in many laboratories around the world to understand the genetics of apomixis and to transfer apomictic gene into the hybrid varieties. Okay, so there is too much research is going on about this apomixis. So you can see here a difference between sexual pathway and apomictic pathway is shown here. Dear student, you can see in sexual pathway, gametophyte embryo sac is there, which is a haploid one that have a haploid polar nuclei, haploid egg cell, haploid synergids, haploid antipodals, and they have a male gamete which are also haploid. For double fertilization will occur, and the particularly this two polar nuclei, haploid egg cell, haploid male gamete, haploid synergid, and all this will occur. And after fertilization you will see there is a seed with the particularly 3 and endosperm and diploid embryo. But when we the apomictic pathway, in this first fertilization will not occur and meiosis will not occur, so diploid embryo sac will be obtained. And Pseudogamy means complete fusion is not happening here. Gamete can enter, but gamete cannot fuse. So diploid nucleus are there. Okay, and parthenogenesis is happening. So if this particular diploid embryo and diploid one is there, if this diploid will undergo particular division and form a what embryo and phi because here meiosis has not happened. See, actually fertilization also does not occur. Here one they have shown some of the pseudogamy, but actually without pseudogamy also we can show this particular apomixis. Okay, so just don't go with this fertilization part. This is somewhat more detailed thing. Without this also you can understand if this particularly thing is a diploid egg cell is if diploid egg cell produce directly diploid embryo, then it is diploid parthenogenesis. Next up after apomixis we are going to talk about parthenocarpy. Parthenocarpy is the production of seedless fruit, and here the fruit production without fertilization of the ovary is called parthenocarpy. So these fruits do not possess seeds because fertilization is not happening, and this occur in case of the grapes, banana. You can see the seeds are absent in them. So parthenocarpy may be natural or induced. It can be induced by some hormone or it can be natural. And this phenomena is applied for the commercial production of seedless fruit. Now, dear student, it is known that seed are considered as one type of irritant. Actually, human being is becoming too much lazy day by day. So we are considering seed as also one type of irritant. So if we get some fruits are there in which seeds are not edible part, then we have to remove them. So there are industries we provide a fruit by removing them and we get a direct things, so that seedless fruit can be generated by particular this phenomena of parthenocarpy. Then one more phenomena which we are going to study here, that is polyembryony. And polyembryony suggests us the presence of multiple embryo within a seed. And this multiple embryo are formed within a seed. Then how they are going to form, that we need to study. More often, as in many citrus and mango varieties, some of the nuclear cells surrounding the embryo sac start dividing and protrude into the embryo sac and develop into embryo. We have studied adventive embryony. If nucellus and integument form a direct embryo, then it is known as adventive embryony or sporophytic budding. And same occur in case of citrus and mango. The nuclear cells start dividing surrounding the embryo sac and form multiple embryos. So in such species, each ovule contain many embryos. So because of the presence of many embryo, occurrence of more than one embryo, it is referred as polyembryony. If we take out some of the seeds of orange and squeeze them, observe the many embryos of different size and shape, particularly found to be seen in this seed. If you see, this is a diagram of polyembryony in citrus. In this citrus, dear student, you can see that this is a normal embryo. Okay, but this is a nucellus region. This is the integument. From nucellus, this embryo are produced. So these are known as nucellar embryo. Here, this is the endosperm. Okay, so such a way, if nucellus give rise to many embryo apart from the normal embryo, then it is known as presence. It is known to be seen with the many embryo, like we have discussed in adventive embryony that it has a 2 to 40 embryo. Okay, and this is showing a polyembryo in mango as well. You can see multiple embryo are found in this. So you can see here that it is showing the presence of multiple embryo. Okay, apart from that, the polyembryo is also of several types. And in this polyembryony, first polyembryony is a particular potential polyembryony. Now in potential polyembryony, what happens? If suppose this is a particularly ovule, and if its ovule has, actually this is found in case of gymnosperm. So if gymnosperm, this polyembryony is found, then they have one integument and many archegonia. And this archegonia has an egg cell. Suppose if there are 2 archegonia or 5 archegonia or as many as possible, you can see, if this is a 2 archegonia, then one male gamete fertilized with this, another male gamete fertilized with this egg cell. If this particular fertilization occur, this is going to produce a zygote, and this zygote will form one embryo. Same way here fertilization also occur, it will produce another zygote, and that is going to form another embryo. So same way, if more archegonia is there, they are going to produce their separate embryo. And in such a way, multiple embryo can be produced. Here, potential embryony is showing that one egg cell is fertilizing with the one male gamete forming a one zygote, and that one zygote forming a one embryo, which is a normal event. But here multiple egg cell are there, so multiple embryo are observed. So it is based on the number of egg cell or number of archegonia, so it is known as potential polyembryony. Then as much as number of egg cell or archegonia are there, as much embryo can be produced. So it is polyembryony. But this potential polyembryony is not true polyembryony. Why it is not true polyembryony? Because if multiple embryo arise from the one zygote, then we can say it is true. Multiple embryo arise. But here, one zygote is forming a one embryo. One egg cell is producing a one embryo. The normal condition is maintained, so it cannot be considered as a true polyembryony. But the next one is we are going to talk about the true polyembryony. That is the second one. That is a cleavage polyembryony. In cleavage polyembryony, what happens? You will see that suppose there is only one egg cell. And with this one egg cell, another male gamete come and fertilize. And fusion occur of a male and female gamete, and zygote is produced. Now this zygote undergo division and form initial embryo. This embryo get divided and produce multiple embryo within it, like embryo 1, embryo 2 and embryo 3. Just I am naming for understanding. So if such a way multiple embryo are produced from the one egg cell or one ovule, then it is known as cleavage polyembryony. Okay, so this is the correct thing, that one zygote is giving rise to multiple embryos, so it is known as true polyembryony. And another third type, adventive embryony or sporophytic budding, that we have discussed in case of okay, that is seen in case of the, we have studied it in case of apomixis. Okay, so now I hope you are clear with this part of polyembryony as well. So let's talk about some of the questions. Production of seeds without fertilization is called. You know that this is apomixis. Okay. Apart from this, parthenogenesis is that parthenogenesis is formation of embryo from the egg cell. Then it is parthenogenesis. If the egg is diploid, then it is diploid parthenogenesis. Diploid embryo will be produced. If the egg is haploid, then diploid embryo will be produced from haploid embryo will be produced from the haploid egg. The polyembryony is multiple embryo. Parthenocarpy is a seedless fruit. The fruit production without fertilization of ovary is called parthenocarpy. And parthenocarpy, if the fruits are produced without fertilization, that fruits are going to be seedless. Occurrence of more than one embryo is called polyembryony, and we have studied the types. Okay, dear students, so in today's lecture we have discussed detailed about the seed, the different dicot and monocot seed. We have studied one of the important thing about the aileron layer in that. Then we have studied the seed viability. Okay, then we have studied apomixis, parthenocarpy, parthenogenesis, and polyembryony. Okay, dear students, so I hope you have understood this in detail, and this is all about the theoretical part of sexual reproduction in flowering plant. Now I will take one more lecture, and in this we will discuss all the previous year questions which was asked in Neet. Okay, so join me in the next lecture. We will be having a detailed understanding of all the previous year questions. Till then, thank you. Have a good day.